Today, the study of human genetics focuses on DNA as the basis for our understanding of heredity and for the location of genes. Our understanding of DNA and our ability to study it in the laboratory have had significant impacts in many areas of society, not simply in forensic science, but also in medical research, commercial ancestry testing, and even in anthropology. Before DNA came to play such an important role, however, there were very few other physical or biological traits that scientists could rely upon to investigate human genetics. The blood groups, ABO, as well as the numerous other blood group systems that were subsequently discovered, were particularly important in this regard. And for much of the 20th century, serology, the scientific study of blood serum, played an important role in advancing our understanding of human genetics. Before the rise of DNA, fingerprints were also recognized as a potentially valuable focus of genetic research. Much like the blood groups, fingerprint patterning stays the same throughout one's lifetime and is not affected by one's environment, something that differentiates it from many other human physical traits. Over time, researchers confirmed the role of genes in influencing certain aspects of fingerprint patterning. For example, during the 1950s and 1960s, British geneticist Sarah Holt confirmed the significant role of genes in determining one's total finger ridge count, the number of ridges that appear on the fingerprints of all 10 fingers. This value is obtained by counting ridges on each fingerprint, as you can see here, and then adding them up for all of the fingers. Of course, the well-established fact that the fingerprints of identical twins are different from each other shows that even if genetics plays a role in influencing the patterning of friction-ridged skin, other developmental factors not directly influenced by genes are also at work in determining the final form that these patterns take. This is especially true for the ridge characteristics, or fingerprint minutiae, that are used in the examination and analysis of latent fingerprints. To be sure, scientists like Sarah Holt were less concerned with forensic identification than they were with achieving a better understanding of human heredity. In this sense, studying the genetics of fingerprint patterning was more relevant to academic science than it was to the field of latent fingerprint evidence. Nonetheless, even today, it is not uncommon for textbooks and other works on fingerprinting to at least mention that fingerprint patterning is partially genetically inherited. Even if it is not practically useful for forensic identification, this fact nonetheless forms part of the foundation of scientific knowledge on which this identification technique is based.